Well, and the other exciting, another couple of exciting things we have today are two award presentations, and the first that we will do today is the G. Evelyn Hutchinson Award, and this award has been presented annually since 1982 to recognize excellence in any aspect of limnology or oceanography. The award is intended to symbolize the quality and innovations toward which the Society strives, and to remind its members of those important goals. In lending his name to the award, Hutchinson asked that recipients be scientists who had made considerable contributions to knowledge, but also those whose future work promised a continuing legacy of scientific excellence. If you look at the list of illustrious awardees over the years, you can see that we have been very successful, I think, in recognizing people who hold exactly those qualities. It's a really special pleasure for me to introduce our awardee for 2008, Alice Alldredge. I think I first met Alice at the 1999 Santa Fe meeting, or maybe slightly before that, being sort of a limnological kind of guy. But she gave and I don't know if any of you remember that, but she gave a presentation at that meeting that I think is amongst the most inspiring talks that I've ever seen. It was magnificent, and I am very lucky that I have a copy of that presentation on video and have shared it with graduate students. And if any of you would like to see this sometime, let me know. It's absolutely wonderful. She currently, though, is chair of the Department of Ecology, Evolution, and Marine Biology at the University of California at Santa Barbara, and has been there since 1976. In looking over her CV, I noticed that she sort of grew up near where I grew up. Also, she got her BA, or at least worked at Carleton College, got her BA from there in biology in Northfield, Minnesota, and a PhD from UC Davis in ecology. There was a NATO postdoc in Australia after that, and she is a recipient, as you can imagine, of many well-deserved honors and awards, including early on in her career NSF Graduate Fellowship, NATO Postdoctoral Fellowship. She has been awarded the Bigelow Gold Medal in Oceanography. She's an elected member of AAAS. She has received a University of California, Santa Barbara Distinguished Teaching Award. She's an elected Fellow of AGU, is listed as in the Web of Science's most cited researcher list in the top 0.5% in her field, and AGU Sverdrup Lecturer, many important honors. Her first publication was in Science in 1972, and set the stage for a great deal of wonderful work in the future. Not bad to start out with a science publication. Large number of publications in the best journals in the field. The foci of her publications have included, and I'm sure she will review a lot of this, some of this work today, and do it better than I, of course. But the ecology of appendicularia, diverse plankton groups, really a staggering array of wonderful work. Field observations of the plankton, grazing ecology, vertical migration, detrital processing, future of science teaching, and some wonderful philosophical works. Of course, marine snow, ecology of floating diatom mats, nitrogen fixation, bacterial processing and production, process of flocculation, TEPs and DOC processing, and et cetera, wonderful and inspiring work. Alice has also given great service at the national and international level, as well as a wonderful service to our society. She has worked with NSF on the Executive Advisory Committee. We worked with JGOF Steering Committee, Coastal Ocean Processes Steering Committee, many NSF panels, of course. And I remember in her talk in 1999, she said that one of the things that happens to us as we do more science is we simply do more judging, I think is what she said, which was lovely. Ocean Studies Board, NRC, and so on and so forth. You can read them there, and her service to our society has been exemplary and much appreciated. ASLO board member at large, ASLO Budgetary Planning Committee, ASLO Committee on the Future of ASLO, and chair of the ASLO Nominating Committee. The quotations from her nomination and her letters of support were spectacular, as you'd expect. I list a few here. Dr. Alldredge is a scientist whose work has deeply enlightened us, without whom we would have a far more primitive understanding of carbon flux below the photic zone. She's an outstanding spokesperson for science and the role of science in society, helped to transform our understanding of the pelagic zone and empowered a new generation of scholars, something that's very, very important for each of us to do. Brought together the honorable traditions of the naturalist and the technology of modern the modern scientist. She's a pioneer, a truly original scientist, and one whose role and work commends her for this prestigious award, and it is a special honor to introduce Alice Alldredge and Sybil Seitzinger, who will present her with the Hutchinson Medal for 2008. Alice.
So the Hutchinson Award is the award itself is very beautiful and interesting. It comes in a case like this, and there's actually a medal inside. So Alice, on behalf of ASLO, it's a great honor to present this award to you, the G. Evelyn Hutchinson Award, for your work on marine snow, which has and continues to revolutionize our understanding of particle flux and carbon cycling in the sea. Congratulations.
Thank you so very much. So is this on for you? I'm speechless. I can't tell you, really, words can't convey how honored I am to get this award. I've been a member of ASLO for 35 years, and it's my chief society. It's my intellectual home. You know, this is where I come when I really want to have fun and think and interact with colleagues. So to get this extremely prestigious award and be in the incredible company of the other people who have gotten the Hutchinson Award is just a tremendous honor. So thank you. Thank you to ASLO, to the selection committee, and to the nominating committee. I was especially moved that my nominators were some of the students that I've mentored and loved through the years. So thank you very much. And we've got to move this to the beginning, I think. Somehow it got at the end. All right, so they've asked me to talk about my research, and I thought I would talk about marine snow, which is really the thread that has tied all of my research together. So I'm going to talk about the role of marine snow in gels in the ecology of the sea. But before I do that, I want to acknowledge some of the many, many people who have helped me. You know, I'm up here by myself, but I feel like there's really an army of colleagues and students who are standing behind me for this award, and I wanted to acknowledge them. Especially some of my mentors, Gary Wagenbach at Carleton College, who helped me learn how to do research as an undergraduate, and Art Bykema, who introduced me to zooplankton when I was an undergraduate in the Boundary Waters Canoe area near Canada in Minnesota, and who really got me started in plankton. And then Bill Hamner, my major advisor at UC Davis. I realized also when I was at UC Davis, my main colleagues were freshwater people, Charlie Goldman's group, Pete Richardson, Zach Powell, people like that who really taught me what I knew about aquatic systems, and they were primarily lake people, so I still have a lot of roots there that I enjoy. Also, I've had a wonderful number of postdocs, students who have just been phenomenal. My techs, especially Chris Gottschalk, who was with me for about 15 years, who I published a lot with. And of course, my family. This is my son up here, Reno, up in the corner, who's done some work with me as well. And then the many, many colleagues. Oceanography is such a collaborative field, and I have so many colleagues I've worked with, more than even I can list on this sheet. I wanted to especially mention Mary Silver, who's been a really close friend for many years, Uta Passau and Bruce Logan, who collaborated with me in the discovery of transparent exopolymer particles, probably the most exciting part of my career, actually, and Eric Hartwick at ONR, who got me started thinking about the physics of marine snow. As a biologist, that was pretty new to me, so I really appreciated him getting me off on another tangent. So thanks to all of them who have helped so much and really shared this award with me. So with that, let's talk a little bit about marine snow. The Japanese scientists who went down in submersibles in the 1950s first used the term marine snow because, as you can see from these pictures taken in situ, what they saw when they first went down was what looked like a snowstorm, even like the photo here on the left of coastal California, like a blizzard. And they named it marine snow from a very inspiring quote from the classic book, The Sea Around Us by Rachel Carson. And she said, "When I think of the floor of the deep sea, the single overwhelming fact that possesses my imagination is the accumulation of sediments. I see always the steady, unremitting downward drift of materials from above, flake upon flake, layer upon layer, a drift that has continued for hundreds of millions of years that will go on as long as there are seas and continents. For the sediments are the materials of the most stupendous snowfall the earth has ever seen." I love that image because it's really real. Marine snow are visible particles. We think of them as particles greater than 0.5 millimeters, but they're visible like snow, like real snow. And so when we see them in the ocean, they do in fact look like a snowstorm raining down onto the deep ocean. As you can see from this picture, marine snow has a lot of different components. These are some micrographs taken by Mary Silver, and you can see there's fecal matter in some snow, lots of diatom frustules in others, a lot of sort of unidentifiable goo, which I think is actually copepod fecal matter, and then a lot of different organisms, especially ciliates and a whole microbial loop community you can see right here. So it's a very diverse group of organisms and particles that make up marine snow. But really, despite its heterogeneity of composition, there are really only two pathways by which marine snow is formed. And the first pathway, at least in the ocean, the first pathway is a de novo production by marine organisms. Many of the zooplankton produce webs, feeding webs such as the one here shown for pteropods or larvacean houses, which are formed de novo by the animals and then are released or abandoned and form then some of the marine snow that we see in the ocean. The other pathway is also very common in lakes, and this is a pathway via coagulation. We take primary particles like phytoplankton, fecal pellets, microaggregates that might be formed by abiotic processes such as dissolved organic matter absorbing to bubbles and then those bubbles dissolving. A variety of different kinds of particles that can collide together and then stick together to eventually form this material, these aggregated particles that we call marine snow. And when we look at coagulation processes, we see again marine snow that is essentially fractal in nature and has a lot of different components. Diatoms are a major source of marine snow. Here's a diatom aggregate. Often blooms will flock up and sediment out overnight. Here's another marine snow particle with some euphows and fecal pellets in it. So the coagulation process does produce a lot of the marine snow that we see both in freshwater and in marine systems. What I'd like to talk to you about today is, since this is a general talk, I thought I would talk about some of the ways that these particles have important ecological roles in pelagic systems. So I'm first going to talk about them as hotspots of biogeochemical activity, how they're enriched islands in the pelagic zone, talk about food, how they're consumed by zooplankton and fish, also about vertical transport, which is probably one of the places where the most study of snow has taken place, because this is the kind of material this is the major form in which material sediments to the deep ocean. We'll talk a bit about how these particles and gel particles that help glue them together form microscale habitats in the environment, and finally talk about marine snow as an economic nuisance very quickly. So I'm going to be really highlighting just a few things. This is a huge topic, and I just want to make a few kind of points as we go along to help you understand how these particles might be significant. So first, hotspots of biogeochemical activity. Marine snow is really, of course, very enriched in particulate organic matter. It's particulate after all. And when we look at the contribution these large particles make to total particulate organic carbon in the water column, which is what this column right here is showing us, sometimes as much as over 60% of the total particulate organic carbon in coastal oceans can be tied up in marine snow, in aggregates that are larger than 0.5 millimeters in size. Even in places where they're not blooming, such as here in Monterey Bay, we can get around 9%. These particles, however, are rich in carbon. I tend to think of them as sort of candy bars. Normal phytoplankton would be maybe a carbon to nitrogen ratio of 3 to 6 or so. And these marine snow particles tend to have much higher carbon, but they do have some nitrogen, so they're kind of like Snickers bars, and they've got a little bit of peanuts in them, something to make them useful for organisms that might feed on them. They're also incredibly enriched in microbes, as we've mentioned before. And in fact, snow is really a microbial loop community within the interstices and attached to the particles that form the aggregate itself. And you can see in this diagram, a diagrammatic representation of that. We have phytoplankton, a lot of phytoplankton in marine snow that's very active photosynthetically, producing dissolved organic matter that then supports a whole microbial loop, bacteria, flagellates, other kinds of protists, and of course lots of interactions with the outside environment through various colonization and immigration and diffusion within and without the particles. So it's really a very complex community within these particles. And because of that, we see a lot of enrichment of organisms within marine snow. There are just a few things on this slide that I wanted to point out, and all of the sources, there are a lot of sources for this data. Phytoplankton can be up to 10 to the 5th times more concentrated on marine snow than in an equal volume of surrounding seawater. So greatly enriched there. Bacteria up to 1,000 times, right here. Nutrients also up to almost 1,000 times. And ammonia and nitrate are particularly enriched sometimes in marine snow. And even dissolved organic carbon can be 20 times more concentrated within the interstices of marine snow aggregates than it is in the ambient seawater around the particles. So we've got a lot of enrichment on these particles that support a lot of this biological activity. There have been quite a number of studies of growth rates, photosynthesis on particles, all kinds of different biological processes. I just wanted to illustrate this point with one particular study that I did with Dave Smith quite a long time ago now, where we looked at the turnover of amino acids on marine snow. And in that particular study, even though the turnover of carbon was on the order of hundreds of days, some of the amino acids on marine snow were turning over very rapidly in under 2 days, different kinds of flocks over here. So basically, the material that's very labile on these particles does get taken up and utilized very quickly and turned into microbial biomass. The organisms are chewing it up and using it rapidly. Because these materials, especially things like nutrients and dissolved organic carbon, are concentrated in the interstices of snow, these particles, as they sink, can actually leave a signature in the water as they go down. This is some work, not my own, it's by Thomas Kierbo and George Jackson, and they modeled a particle that would not be moving, modeled the diffusion of, say, dissolved organic carbon or a nutrient out of the particle. But as the particle begins to sink, and as it sinks faster and faster, which is what these cartoons here represent, we get a trail coming out of the particle of, say, high dissolved organic carbon or high nutrients, which organisms then in the water column can actually utilize and pick up on. We might think that diffusion would be really, really fast, and such trails might not be very important in the environment, but in fact, in the mid-1970s, Hamner and Hamner showed that suggested shrimp are actually adapted to find such trails. They dropped fluorescent dye-soaked crushed shrimp down into a tank, and these little shrimp would find the trail and they'd trace it down exactly all the way to the food source. So the fact that some organisms have evolved to actually find these trails is a sign that in fact marine snow does leave us, and other kinds of trails such as copepod pheromones can leave an actual detectable trail in the water for a long enough time that organisms can find it. Okay, the second area that we find marine snow being important is as a food source, and there's actually not a lot of information about this particular area. Marine snow is consumed by a variety of different kinds of zooplankton and fish. It's fed upon by myctophids, some midwater fishes. Many benthic communities have fish that will feed on marine snow. Blacksmith, for instance, in kelp forest, come up to the upstream end of the kelp forest and pick up marine snow. And we also get a lot of zooplankton feeding on marine snow. Here's an example. This is from work of one of my students where we fed euphausid adults and larvae, and down here, Calanus pacificus, a copepod, marine snow, and then looked at the fecal pellet production. So what we're seeing here is sort of the fecal pellet, micrograms of fecal pellet per in this case, euphousad per hour. And this is snow plus seawater, and here we have just seawater without snow. And you can see that the animals are in fact consuming a lot of snow, consuming a lot more food than they would in regular seawater. Same here with our larvae, and the same down here with our copepods, so that the FSW means filtered seawater. So they're consuming a lot of more food when snow is present. We, however, don't really know what the contribution of marine snow is to the total food supply in places such as the deep sea or even in the surface waters. It's a very open area at this point. The third place that marine snow is quite important in the ecology of the pelagic zone is with vertical transport. Most of the material that is sedimenting down to the seafloor or into the deep ocean is doing so as marine snow. In some places it's fecal pellets, but it's most places marine snow. And in fact, marine snow is the main material that is captured in sediment traps. Why then, what's so special about marine snow that it is going to be a major transport mechanism? Well, first of all, it's very high, as we've mentioned, in a particular organic matter content. Here's just the dry mass of the particles versus aggregate size down on this axis. And as we would expect, bigger particles have higher mass, but they have a high particular organic matter content because of their large size. Marine snow also is relatively abundant. This is some data from coastal waters where we get down 40 or 50 meters and deeper, maybe 20 aggregates per liter, much more abundant even up in surface waters. And in the deep ocean, we're going to get much lower, maybe 1 or 2 aggregates per liter. But still, this is a fairly high abundance when you consider that this material is sinking down as well. And that leads to the third component that helps make this an important transport mechanism, and that's that it sinks very rapidly. This is the results of a fun study I did with Chris Gottschalk, my tech. We went out and shot a little bit of dye right below aggregates in situ, and then very carefully pulled away from the aggregates and actually timed their sinking in the ocean. And when we did that, we get this kind of a relationship between size and sinking rate, a lot of scatter because of variety in the particles, but basically there is a relationship. Even though aggregates sink on the order of tens, on the order of 50 to 100 meters a day is more typical, it's still going to take them 40 to 80 days to reach the ocean bottom. So you can see that a phytoplankton cell sinking only a few meters a day at most is going to be long gone before it reaches the bottom. Marine snow, however, there's still a little bit left 80 days later, and they can finally hit the bottom and contribute to the sequestration of carbon in the deep ocean. And finally, as these particles are sinking, the last thing that makes them important in transport is that they're very strong and resistant to fragmentation. And this part's a little longer because I like it. It's kind of fun. I thought you might like it too. One of the things that I've looked at with Tommy Dickey and Tim Granada was to look at the strength of marine snow, the physical strength of it. And what we did is we had a meter-cubed tank in the lab with a grid, an oscillating grid in the bottom, and that grid would go up and down like this. And as you move away from the grid up toward the top of the tank, the amount of shear stress or the energy dissipation decreases. So right near the grid, it's really, really energetic, and as you move farther and farther up in the water column away from the grid, it's less and less energetic. So what we did is dropped particles down into the tank and saw at what level of energy they began to break apart. And to our surprise, many of the particles were extremely strong. In fact, particles that are miscellaneous debris, primarily fecal matter, aggregates that are formed of fecal pellets, larvacean houses, there's a bacterial flock. These were so strong that they went, 100% of them went right through the grid. They didn't break up at all. And among the more fragile aggregates, diatoms aggregates formed from Contoceros and Nitzschia, about half of those did break up before they hit the grid at energy dissipation rates less than 1. That's an energy dissipation rate about what you'd see in a rapid tidal current, for instance. And so let me show you a little bit of this. Here's an example of a really strong aggregate, and this one's going to bounce like a basketball right from the grid, and it's not going to break up. So it's gone down below the grid. You can see the grid bouncing here. Now it's coming back up, and it's going to come up yet one more time. There it is again. Still refuses to break up. So some snow is very, very strong indeed. That was a larvacean house. Here's a more typical fragmentation process. This is a diatom aggregate, and I should point out that this scale over here on the left has nothing to do with the size of the aggregate. This shows us the distance away from the grid it was. It was taken by a second camera. So watch this aggregate, and you'll see it begins to disaggregate. There's still quite a lot of turbulence you can see around this aggregate before it disaggregates there into 2 particles, and now it's going to break up again into several others. But notice that in the disaggregation process, most of the daughter particles are still pretty big. They're still in the marine snow size range. There's one other way that particles break up, and that's from animals. And one of my students and I had a lot of fun working with Euphausia pacifica. What we did was and with Jeanette Yan as well, we tethered a euphausid and we dropped marine snow past it. And as you drop the marine snow past the animal, it gets sucked into the pleopods, and you can see here is the pleopods are going to break it up. Here we go. And so it's like these big animals are moving like tunnels. They're making tunnels through the water as they move around, not just krill, but salps and all the other larger animals. And as they move around, any particular kind of marine snow that might be near them could get broken up. So this may be an important mechanism to break up marine snow as we go deeper and deeper in the ocean. The chances of a marine snow particle coming into contact with an animal increases with the more time that snow particle spends in the water. So I guess the take-home message there is these particles are strong, but they can be broken apart by really high shears, such as we might see around the pleopods of animals. Okay, back to the ecological significance of these particles. The fourth one I wanted to address was the aggregates and the gels I'm going to speak about provide really important small-scale structure. We tend to think of the ocean or a lake as water with organisms, microscopic and macroscopic organisms floating and swimming in it. But in fact, that's not really what it's like. It's actually full of surfaces. Our pelagic systems are full of particulate surfaces that provide refuges and places for organisms to hide and to rest upon, for that matter. In fact, you can see here in the pictures I've been showing with quite a number of these slides, a couple of copepods sitting on a marine snow particle. This is Oncea, and it's the genus Oncea, and it's actually able to scrape. It's adapted to scrape particles and food off of particles like marine snow. It's its whole way of feeding. I didn't realize how important the structure was in the environment until one time I was out making movies in the open Pacific. I was doing research and we were making movies on the side. We had some really, really bright lights, and as we shine these lights into quite a ligotrophic ocean water, all of these sort of cobwebs and sort of structures appeared to our eyes. It was as if the water had a real physical structure that was totally invisible to us until we shone the bright lights on it. And that material turned out to be the subject of another area of my research, which was that of gel particles. It turns out that the ocean is filled with gel particles, large disks and strings and globs of gel material. We call it TEP, or transparent exopolymer particles. They're really a subset of what many of you know of as EPS, extracellular polymeric substances, but we called TEP a different name so that we could make it really clear that these gels were particles in their own right that could contribute to aggregation processes, and rather than being coatings on other kinds of structures, coatings around diatom cells or biofilms. TEP is formed by exudates from phytoplankton and bacteria. Here's a photo by Gary Lepard showing these polysaccharides coming off the surface of a cell, and then almost immediately they begin to form cation bridges with the cations in seawater, and they form these microfibrils, which then continue to aggregate up and form large gel particles, which we can see here, and strings and globs, which are made visible with Alcyon blue, which is a stain for acidic polysaccharides. TEP are about as abundant as we find phytoplankton, and they form the matrix of marine snow, of marine aggregates. They're the glue that really sticks the relatively unsticky marine particles, other marine particles, together. Phytoplankton and the like are not very sticky, and so this material sticks them together. Here's an aggregate made in the lab, and you can see all this Alcyon blue, glue-like material sticking diatoms here together. And here's a natural marine snow particle down here with big globs of TEP actually participating in the aggregation process, sticking diatoms together. TEP can be up to several hundred per milliliter, ranging in size from 3 microns to hundreds of microns, on average around 10 microns in size. So we can get a lot of TEP in the water, and this can really help glue the material together. These gels, and of course our marine snow itself, then provide refuges and microhabitats for bacteria and microbes. To illustrate that, here's a string of TEP, which you can see, and double stained the exact same string with DAPI. And normally we might think, and here's some free-living bacteria, but notice how many of the bacteria are really associated with these TEP particles. Here's a TEP particle here, and you can see it outlined in the bacteria. They're on it as well. So clearly these gel particles are important structures that are providing a lot of heterogeneity, a lot of microstructure to the microbes that live in aquatic systems, and providing them on scales of a few microns up to as much as a meter, because there are larvacean houses that are a meter across. So it's an important aspect. This microhabitat that these particles provide is an important aspect of the importance of these aggregated particles. Finally, marine snow is an economic nuisance. This occurs primarily in the Adriatic Sea, where we get huge snow forms from some diatom blooms that occur there sporadically every few years, actually. And that snow floats up to the surface and forms huge scums that go hundreds of kilometers in extent. These are, down here, are beach umbrellas along the coast of Italy, these little red dots. And this is a billion-dollar tourist problem for the Italians when these scums are formed. Eventually this stuff sinks down and smothers the bottom and kills a lot of the material in the bottom of the Adriatic Sea. Parts of the Mediterranean and the Adriatic are the only places we see this kind of event. So in summary, marine snow are aggregated particles greater than 0.5 millimeters, and they're an important part of marine ecosystems. They form particulate islands that are enriched 1 to 5 orders of magnitude in materials and organisms. Marine snow is consumed by zooplankton and fish, and its large size, its abundance, its rapid sinking rate, and its resistance to disaggregation combine to allow marine snow to reach the deep sea before complete decomposition. And the formation of marine snow requires transparent exopolymers, these TEP materials as glue. Marine snow and TEP provide microenvironments and refuges for microbes in an otherwise relatively homogeneous pelagic environment. This field can go a number of places. There's a lot of things we don't know yet. I think some of, just a handful I threw out quickly, the role of ballast in snow sinking. That's kind of a hot topic right now, the role of ballast in flux. Microbial processes on marine snow habitats, especially in the twilight zone, in the oxygen minimum zone. Didn't have time to show you the data, but marine snow can get nearly anaerobic, and as it sinks through an oxygen minimum zone, there may be microbial processes that could occur there under those conditions that we wouldn't normally find up in the water column. The importance of marine snow as a food source is still relatively poorly known. We need to quantify decomposition processes a lot better. And finally, the diurnal patterns of aggregate formation are really interesting. I'm not sure how marine snow is forming. When you look at a diurnal pattern, it's broken up at night to some extent by the zooplankton migrators that come up and that both physically break it up and eat it. But we also find a lot of diurnal patterns in the formation, lots more snow in the midday periods than we see later on. So there's a lot of interesting questions with regard to formation. So I hope that this has stimulated your interest a little bit in marine aggregates, maybe in lake aggregates if you're a lake person. Some of the early work on aggregates actually was done in lakes, in Lake Tahoe by Hans Perl back in the early 1970s. So there is a history of lake snow as well. And I think of course, I love this topic. It's a really fun one for me, but I hope I've stimulated you a bit with it. And thank you again so much for this award. It's a tremendous honor. Thank you. Thank you. Sure.
I'm sure there are a few questions for Alice. Who would like to begin? Could you use the microphone, perhaps? Thank you.
I might come down here. Is that okay?
Sure.
Yeah, it's right up there. Do you want me up there? No, it's yes? Okay. Sorry. Okay.
To me, a lot of this was actually new, and I'm wondering, organisms like diatoms that are living, do they get trapped in this stuff, and is that something they have evolved?
Yes.
Because it's bad for them, I guess, to sink out.
Well, actually, diatoms are really interesting. Diatoms, I've done some work with diatom flock aggregate or diatom bloom aggregation, and Victor Smetacek had a paper in 1985 where he predicted that diatoms would aggregate up and sink out overnight. And then later I actually documented and saw that. And it turns out that the diatoms have extremely high production photosynthesis in their flocks because there's a lot of nutrient recycling and they get more nutrient replete. And what they do is they have this high photosynthetic rate, get nutrient deplete from the microbial loop organisms there, and then they shed cells off into the water. So you could almost evoke a kin selection kind of argument that some of the cells are helping the others stay up in the water column by sinking out. But Victor also believed that sinking out of diatoms was an important adaptation to get their oxospores down into the sediment, and then they'd resuspend later when upwelling events happened again. So there's a lot of benefits to diatoms to be in aggregates, actually, and a lot of photosynthesis happens there. Yeah, thank you. Any others? I can't see that. There's okay.
Yes, right here.
Congratulations. And has your research given you any insight into how humans might manipulate aggregation processes to accomplish engineering objectives, maybe to manipulate the carbon cycle or wastewater treatment processes?
Wow. I've thought about those nuisance blooms. I bet the Italians are busy trying to scoop up that material and turn it into biofuels. Wouldn't that be just a lot easier than getting it out of the ocean? Just use a bulldozer on the beach. Wow, what happens in sewage plants, of course, does produce a lot of flux. It's very similar to what's in the ocean, and a lot of some of the early work on flocculation is from those systems. Actually, I really haven't thought much about that. I'm not sure. Have you? Do you have any ideas? You study it also.
I haven't really started to think about that too much.
Yeah. Certainly.
Interesting to consider.
Yeah, it is. Certainly, adding polymers, of course, has done a lot to increase flocculation, and that's what's doing it in the ocean as well. These exopolymer particles are really involved in flocculation, so we might be able to seed, I suppose, some blooms and get them to sink out more rapidly, which might help a little bit with carbon sequestration. But I think that again would take a lot of, just like iron fertilization would take a lot of fuel just to get out there and do it, and it'd have to be done over long periods of time, so probably not very practical. Yeah.
Alice, thank you for all your work. I'm a big fan of it. But why 100 meters per day? As you know, I started doing oceany stuff and then moved more into muddier stuff. And we get the same kinds of settling velocities for flux in a whole range of environments. And that's fascinating because it has that's a fundamental biogeochemical parameter for ocean models, that this stuff sinks at 100 meters per day. So do you have any?
They're very fractal and incredibly porous. Some of the diatom aggregates are 99.9% porous, which is more porous, I think, isn't it, Paul, than most of the small, more mineral aggregates that we get in estuaries and the like. So
yes, but they get smaller, and then they tend to be smaller and sink at the same rate.
Yeah, yes. Yeah, so compacted but smaller.
Small,
so
dense stuff sinks at the same rate as the large diatom wall.
Yeah. Some snow.
It doesn't seem to change. So
No, it's true. I think that the material that it's made of is really important. Diatom flocks sink pretty rapidly. I've seen some pretty fluffy snow that doesn't sink as rapidly. Doliolid pellets, for instance, are pretty fluffy and don't sink so fast. Larvacean houses don't sink so fast either. They're mostly water because they have relatively little material in them. So there is some variability. You can get down to 25 or 30 meters a day, and you can get, of course, higher, a couple hundred meters a day or more. But yeah, that's interesting. I've often thought that the variation in marine snow sinking from, say, 25 to 200 meters a day wasn't that great a difference relative to other parameters that affect flux. I don't know if you would agree with that, but
that's my point.
Yeah.
There's not as much variability.
Yeah, exactly. Other things may be much more important in determining the quantity of flux that gets down than the sinking rate.
One final question. Thank you.
How much do you think diatom's gliding ability is playing a role in maintaining flux or creating flux and diatom life on flock?
Diatom gliding abilities?
Yeah.
I'm sure that the diatoms that can glide are gliding around a bit in the flocks themselves. Most of the diatoms that are forming pelagic flocks, things like Cattosaurus and Nitzschia, aren't much like some of the benthic diatoms. They don't glide that much. They're really being collided together by physical processes in the water and then stuck together rather than moving toward each other, rather than having it be sort of something they're involved in the movement with. But flocks on the bottom, that may mean something quite different. I mean, we've heard a lot of talks today about flocks in streams, and there you get a lot of movement of diatoms, and they may be able to help their own aggregation in that sense. Yeah. All right, thank you so much.